So up to this point, we've mostly been talking about proteins and their function in the cell, and we've mostly been focused on that because, again, proteins and enzymes perform most of the chemistry within the cell. But as we know that to synthesize a protein, you need to have some sort of instructions or information. And so this is where DNA comes in, okay? So if we have a cell, for instance, such as a eukaryotic cell, we know that DNA is stored in the nucleus for a eukaryotic cell. For a bacteria, the DNA may be free in the cytosol of the cell instead of enveloped inside of a membrane-bound organelle such as the nucleus, okay? So from there, this houses the DNA, and the DNA is the instructions of how to make all the proteins and other components that are necessary inside the cell. And so you may recall what's called the central dogma. For molecular biology, right? And so what that says is that one of the central tenets of that is that one gene codes for one peptide, okay? Now there are exceptions to this, but it gives us a framework to start talking about what we need to in terms of going from DNA to protein. And so remember that DNA is double-stranded, and then this is going to be transcribed. So the next step of this is transcription. So DNA acts as sort of the master instructions for all the proteins in the cell, and that stays in the nucleus, and it gets protected in the nucleus in a eukaryotic cell. This gets transcribed to RNA, which is single-stranded, and can have all sorts of secondary structure depending on the sequence for the RNA strand, okay? And so again, this is single-stranded for the most part, instead of the double-stranded DNA. And then finally, we go through what's called translation. The cell goes through a process called translation. And this is where codons, again, from the DNA, which is transcribed into the RNA, codons are translated into amino acids. So we have codon tables for that. And that ultimately results in a protein. So this next section that we're going to look at mostly the structures of DNA and RNA, what makes them different to enable their different functions. One, DNA for storage, information storage, where there's a premium on stability, so not having the data be modified in any way, such as by a mutation, or not having it degrade. Whereas RNA is, again, for messaging, so for transcription, and plays more of a messenger role. And in this role, it's not as stable and can be quickly degraded if need be. So this is involved in regulation, in other words, which can help control how much protein is made, and therefore can control the chemistry that this protein or this enzyme is important, that this protein or enzyme mediates. Okay, so first we're going to look at, before we get started on looking at the nucleic acids, which we consider DNA and RNA, looking at the macromolecular structure of this, we first need to know more about the monomers that make up both of these.